is my pleasure to introduce Dr. Elmar Nurmemmedov, who will be talking about cellular target engagement as a new paradigm in drug discovery. Dr. Elmar Nurmemmedov's research is focused on structural biology and drug discovery in the field of challenging drug targets, particularly transcription factors. In the past several years, he has been trained in cancer biology and molecular pharmacology. During his training at Children's Hospital Boston, Harvard Medical School, he led the Drug Discovery Project at Broad Institute for Sickle Cell Anemia. His training in molecular biophysics and molecular pharmacology contributed significantly to identification of early-stage drug candidates. Altogether, Dr. Nurmemmedov aims to continue his scientific career with molecular pharmacology and drug discovery applied especially in the field of glioblastoma. Take it away, Dr. Nurmemmedov.
Thank you so much, Andrea. Thank you for a kind introduction. I'm trying to share my screen here, and well, thanks again for introduction. Thank you for having me here. I appreciate the opportunity. So I will be talking about cellular target engagement as a new paradigm in drug discovery. So my presentation is split into 3 sections. I will first spend 7 slides to talk about cellular target engagement in drug discovery as a paradigm in general. Then I will share with you 2 stories. Beta-catenin in drug discovery. This has been work in my lab for the past several years. And then I will talk about PLK1 drug discovery. This is work we have been doing with our collaborators at University of South Carolina. So here we go. Drug discovery is long and expensive process. We all want safe and efficacious drugs, and that is a challenge. So drug discovery industry is set for this long process, and it's very expensive, takes upwards of 12 years and upwards of $2 billion to bring one drug from the inception, from the drug discovery phase, all the way to commercialization. And there are many caveats. There are a high risk and very high attrition rate. So this is a picture you have seen many times and you're well familiar with. So target engagement happens to be one key reason why drugs fail. So on the right here, you see as a result from a recent study where they show why drugs fail. And one big reason, 52%, is efficacy. And then we have safety. And then there are some other reasons. So efficacy is basically split into 3 subsections: insufficient target engagement and poor pharmacokinetics. Preclinical models did not translate. And again, insufficient target validation data linking it to the disease model. And on the safety side, we have then the mechanism-based toxicity, off-target toxicity, and then basically other medchem reasons. So really the target-related issues come at the forefront of the drug failure in the clinic. So if we could address these issues up front in the early drug discovery, then perhaps we would be much better off bringing drugs to the marketplace in a more safe, secure, and commercially viable way. So the million-dollar questions here for the drug discovery industry are, is your drug really a therapeutic? Does it engage its target inside of the cell? And these are the questions we really want to answer early on in the drug discovery process. Today, we are really good at using the omics data to drive our drug discovery efforts. We use a lot of genomics, we use a lot of proteomics data, and we collectively drive our decisions as we go through design, discover, optimize, and develop stages of drug discovery. And hopefully by the end of that pipeline, then we end up in clinical trials. And what you see here is early drug discovery. And this is the highest risk and the longest chunk of the drug discovery process. So cellular target engagement is a new paradigm that is already shifting the face of drug discovery. Why is it important? Because it's more physiological, it's really target specific, it's disease relevant, and it's responsive to the target in question, and it can really challenge and interrogate challenging therapeutic targets like transcription factors, like proteins, like you would otherwise not be able to work with under artificial conditions. So target engagement has been around for a while, and what's new is in this field is really cellular target engagement. So I have here a diagram showing old technologies versus emerging technologies, and in the vertical axis, cellular technologies versus non-cellular technologies, just to show you a big picture here. So on the upper left side, you see old cellular technologies like Pelago, relatively old, I should say. They are still very much used, but they have emerged some time ago. These are reporter assays, and these are previous enzyme complementation assays. Right bottom side, you see conventional tools like affinity measuring by SPR, crystallization, fluorescence polarization, microcalorimetry. On the top right corner, you will see cellular technologies that are up and coming. These are Promega, NerdBio, and these are newer, bolder technologies that really claim to address the challenges of drug discovery today. And on the lower right side, you will see emerging non-cellular target engagement technologies like micro scale calorimetry, thermophoresis, and the proteomics. So this is pretty much a complete picture of target engagement. And what is really interesting for us is the cellular piece, the upper right corner, which seems to be driving the and addressing much of the challenges in drug discovery today. So conventionally, target engagement works by immunoblotting. It's a very standard, well-established procedure. You have a cell, you expose the entire cellular content to a temperature gradient, and your target protein melts together with all the proteins inside of the cell. And then you remove the unstable aggregated protein, you detect by immunoblotting, and then you see your protein band taper or disappear in the temperature gradient. When you add a ligand, a drug, you expect that ligand to rescue your target protein from denaturation, and then you see a shift, like in here, blue line versus the red curve. And that temperature delta T is indication of target engagement and rescue of denaturation. Advantages of this method are live cells, no modification of target, no modification of compound, no labeling, and you can calculate EC50 and you can rank your compounds using this method. Disadvantages, it's not scalable, it's labor intensive, it's immunoblotting. The newer way of looking at target engagement is by enzyme complementation, and there are a number of companies and platforms offering this. Basically, you have an enzyme, you attach a small piece of that enzyme, this red amino acid tag, to your target protein, and then you express it in the cell. And then when you detect that, you add the larger subunit together with the reagent, and based on the engagement of your with your ligands, you have a colorimetric readout, and that can be very quantitative, and that can relate to target engagement. Advantages are you use live cells, no modification of the compound. This is highly throughput, it's fast, you can rank your compounds. Disadvantages are that you attach a small amino acid target to your target protein. So essentially, that could be anywhere from 12 to 45 amino acids, and that could be a critical point to consider. So with this, I would like to shift gears and show you how we have used target engagement in our previous studies. So in my lab at John Wayne Cancer Institute, currently St. John's Cancer Institute, we have been working with Wnt pathway, particularly beta-catenin. Why is this important for us? It's because it's implicated in a number of cancers, basically. All these cancers, starting from colon cancer, breast, lung, and all the way to glioblastoma and liquid cancers, they're even driven or co-driven by Wnt beta-catenin. On the other side, you have a regeneration side of the pendulum. If you can nicely co-activate beta-catenin, Wnt, you have regeneration of tissues, and this is a field that's emerging and very progressive. So we are on a mission to discover direct inhibitors of beta-catenin, and the pharmacology of beta-catenin has been established for a few decades now. So what's really interesting for us is to discover direct targeted inhibitors of beta-catenin versus inhibitors of the upstream of the Wnt pathway. So direct inhibitors of beta-catenin are considered the safest and most efficient therapeutic approach against the Wnt pathway, because then you do not interfere with the upstream cross-talk with other pathways. So what we have done is we have scanned the surface of the beta-catenin crystal structure. You see here, we have identified red hotspots which have the excess binding energy for small molecules. We have 4 of these allosteric binding sites, and then we have screened virtually and we have docked a library of 200,000 compounds, and they found around 155 compounds binding to various sites. Of particular interest for us was site C, and we happened to discover a compound called C2 that binds to site C. So, this is a zoomed in picture into the site C and binders to that particular site. So compound C2 was selected as an interesting compound, which was fairly selective versus and less toxic versus the other binders. I will fly quickly through some of these slides because my point is show you a bigger picture of how we navigated through drug discovery effort. And then basically we have done a number of biophysical cellular assays on C2, which I'm not showing here. And then once we got convinced about C2 being a genuine hit compound, we have done a medicinal chemistry optimization. Basically, we have branched out into 2 directions medicinally. So basically 2 chemical classes now, 1525, 1766. Each class has more than 100 compounds. And then this was a rigorous effort to undergo and select the right better hits. So this is our screening cascade. We have on the top the screening panel, and then we have cellular panel, and then we have in vitro, in vivo, and then further panels. So essentially, I want you to focus on the top part of this diagram. We have luciferase assays, which are classical in the Wnt field, and then we have thermal shift, basically thermal cellular target engagement assays, and then basically we have other target engagement assays. So we put target engagement assays up in the top of the screening cascade so that we can identify the molecules, the ligand that target beta-catenin early on, and all of the subsequent assays would be dependent on that. And this was fairly successful for us. So we have identified 2 compounds, 1525, 1766, and this is a top flash report or assay profile of these two compounds. So as you can see, 1525 will inhibit the luciferase signal, whereas it will not inhibit the constitutively expressed CMV signal. And similarly, 1766 was inhibiting luciferase top flash signal in a dose-dependent manner. And then cellular target engagement for these two compounds, 1525, 1766. Now, there are two ways you can do target engagement in the cell. The first way, and the classical way, is you take a constant one dose of the drug and you expose that to a temperature gradient, and you will see a shift, like you see here. The second way is basically you select one temperature and then you test a dose range of your inhibitor, and that will give you an EC50 of that compound. So in this case, we chose the first one. So this is immunoblotting showing beta-catenin melting profile with DMSO, with a reference compound, and our three compounds, 1525, 1766. You can see that under DMSO treatment, beta-catenin has a melting temperature around 45 degrees Celsius. Added complex that with 1525 and or 1766, you can see that the melting temperature increases to around 53 degrees Celsius, so 4 degree shift. And on the right side, you see the plot of that. So this was the early indication for us that our hit compounds and optimized hits indeed targeted beta-catenin inside the cell and provided us enough confidence to guide our medicinal chemistry efforts. So we did this process iteratively, and we basically ranked the compounds and we took the best compounds forward. So some more background biochemistry about the compounds, 1525, 1766. They deplete beta-catenin inside of the cell in dose-dependent manner. Dose is not shown here, but it does work that way. So when you treat the cells with these compounds, basically beta-catenin undergoes ubiquitin-dependent degradation. So you sequester beta-catenin, you remove it from the cell, and basically you reduce the oncogenic function of beta-catenin. And in a time-dependent manner, we see that beta-catenin is phosphorylated, meaning that it's primed for denaturation and degradation. And it's very important to show what happens to beta-catenin in the nucleus and in the cytoplasm. And here are the two sides of the cell, the nuclear and the cytoplasmic fractions. Both of the compounds work identically. And you can see that upon treatment in the nucleus, beta-catenin completely disappears. I mean, it's like beyond detection. Similarly, in the cytoplasm, you can see that beta-catenin goes to a negligible level, suggesting that our compounds that we discovered using cellular target engagement method as a starting point really worked. And then we did a lot of mechanism of action studies. We did genetic knockdown of beta-catenin and compared that to the inhibition profile with the compound 1525. And we ran RNA-seq, and we find that the two profiles are almost identical. Just look at the general picture. You will see that the genes that are upregulated and downregulated in the genetic pharmacology and in the chemical pharmacology have a close overlap. And just a final piece about the mechanism of action. Our compounds that we discovered work by arresting cells in the G1 phase, and by FACS analysis, you can see basically that there is an increased peak for that phase, and cells basically accumulate in that G2M phase and then go to apoptosis. And then basically this is a complete pharmacology profile of 1525. You can see that it does interact with purified beta-catenin, and we measured that by MST. And basically, we have then a cellular thermal shift assay confirming that direct binding. And we have in vivo studies suggesting that the new drugs work. And currently, we are considering various drug combinations. And this study is being prepared for publication, hopefully very soon. So the second story I really want to quickly mention you about is the work we have done with our collaborators at University of South Carolina. It's their project. So we just helped them use our expertise in target engagement and help them identify and speed up their efforts for discovery of small molecule inhibitors to PLK1. The challenge here is PLK, there are inhibitors, pan inhibitors for PLKs, PLK1, PLK3. What they really wanted to do is identify selective inhibitors targeting exclusively PLK1. Why it's important? Because PLK1 drives a lot of oncogenic transformation and tumor growth in many cancers, particularly prostate cancers. And this is of prime interest. So what our collaborators did, basically, they have a three-dimensional crystal structure, a model for PLK1. They have identified this cryptic pocket, and they have modeled their small molecules fit inside of that crevice. And based on this model, they have come up with a list of R groups, and they have derived their compounds. And basically, it's a very elegant work done by them. At this point, they asked us, can you help us interrogate all of these compounds and identify the ones that potently and selectively bind to PLK1? And what we have done is basically, at this point, we have run a thermal profile for PLK1 and PLK3. On the top panel, you will see thermal melting profile of PLK1. On the bottom, thermal melting profile of PLK3. So what we did is, this is the first step to doing thermal cellular target engagement. You run a gradient, a heat gradient, and you identify the melting profile of your target protein. PLK1 happens to have a temperature of aggregation of 50 TAC of 56 degrees Celsius, and PLK3 happens to have TAC of 48 degrees Celsius. Why is this important? Because in the next step, we will use these temperatures to interrogate a dose range of the of their candidate small molecules. As a first pass, we have taken their panel of compounds. It's a large panel, so we cannot run a complete dose response for every compound. So what we have done is we have run 3 doses for each compound. This is 0.03, 0.3, and 3 micromolar for each compound. And then basically we look at the stable protein fold change in the cell. And we basically draw a selection baseline. You see it's 1.2-fold. And you will see that here that some compounds perform above that selection baseline. So there's a reference compound BI. There is this compound. There is a compound called 6123. There is a compound called the 6125. And then there are basically 1, 2, 3, 4 compounds that perform above that level. And this is done at 56 degrees, which is the melting point for PLK1. So then we interrogate every compound, selected compounds, in a complete dose response manner, and we look at a binding engagement of PLK1 versus PLK3 versus an internal control like GAPDH. Here you can see that basically this compound, 6125, engages PLK1 in dose-dependent manner. The red line, EC50, 0.61 micromolar. The same compound interrogates PLK3 at a weaker EC50, 3.7. There is great selectivity here, almost 5-6-fold. Another compound, 5915, and we can see there is an identical trend here. PLK1 is engaged at 0.8 82 micromolar EC50, and PLK3 is engaged at a much higher EC50. So, and again, this compound is highly selective to PLK1, and you can see basically the immunoblotting gets more intense with the higher doses, whereas PLK3 is not responding. This is an inert compound, 6130, and you can see that both PLK1 and PLK3 are not responding. So this is good. It shows that the system responds to selective and non-selective or otherwise inert molecules. So then we basically went through the entire effort and we tested some more compounds. This is a reference compound, BI 2536, and this is a pan PLK inhibitor. And as you can see, it both engages PLK1 and engages PLK3. And so basically, the profile of some of our new compounds will be identical to this reference compound. Both PLK1 and PLK3 are engaged. So some other profiles. So we found that this compound, 1691, is also selective to PLK1. As you can see, that it increases. PLK3 is not engaged. And the compound 6208 does not engage none of them in a significant way. So some more profiles, I will pass them for the sake of time. And just my message here is to deliver you the picture of what target engagement method could do for you in the early drug discovery phase, how it could accelerate your efforts and guide your medicinal chemistry and focus your efforts on the right compounds. So basically then we carried the PLK1 inhibitors to the enzyme complementation method. This is another second type of target engagement without any immunoblotting. So basically we confirmed that our hit compounds perform in an identical way on enzyme complementation target engagement platform. This is a more scalable platform which you can use to screen for hundreds or thousands of compounds. And you can identify and rank them by EC50. And again, this could be a very informative way to screen and to prioritize your hits early on. So PLK, two last slides here, bear with me. So we found that our PLK1 inhibitors bind at the interface where the two domains of PLK1 would make dimerization contacts. So this blue dot here is the PLK1 inhibitor that was identified through our, not exactly identified, but confirmed through our target engagement efforts. So basically, our compound is able to block dimerization of PLK1 and in a selective way. And when that happens, the compound is able to lead PLK1 to degradation. As you can see here, PLK1 goes from high to low in dose-dependent manner, and you can also combine that treatment with calcimide, which is a potent microtubule poison. Basically, it will drive PLK1 up. As you can see, it's up. And then you can actually bring it down with the new PLK1 inhibitor. Again, it goes down in dose-dependent manner, showing that the identified PLK1 hit compounds are genuine, they're direct, and they really work inside of the cell. With this, I would like to thank you for your attention. I would like to thank St. John's Cancer Institute, Pacific Neuroscience Institute, and all of the collaborators who have been great and provided this highly intellectual and rigorous environment. Thank you so much. I hope you'll have questions.
Thank you, Dr. Elmar, for your really innovative subject that you just touched upon. And we will open the floor for a few questions or remarks if there are any from the audience.
Should I stop sharing?
Yes. Yep.
can ask me now the questions or during the panel discussion. Either way is fine.
Yep. Dr. Elmar will also be on the panel, as you mentioned, from 4:35 to 5:50. Along with Dr. Andrea, they'll be discussing accelerating drug discovery with AI and breakthrough tech. And so do stick around for that one later today. Otherwise,
I had a quick question. So one of the things that came out in your talk is obviously that you're able to really look at the enrichment of these inhibitors for specific PLK subtypes. My question is, do you also do a broad panel? Because one of the issues with these inhibitors is that off-target effects can have really profound side effects.
Right.
At any point in their process, do you look at the inhibitors and their ability to potentially modulate the whole family of these kinds of kinases?
Right. The mechanism of action studies and the selectivity studies will continue even after you validate their binding inside of the cell. So, and of course, this is a very resource-intensive process. So what our collaborators decided to do is basically they will carry the target engagement platform into various directions. So right now, where they are looking into testing PLK1, the parental wild type, versus various mutants of PLK1. And basically, that will give you a more rigorous approach to the mechanism of action and whether or not there is a true selectivity between various PLKs and various binding domains. And one possibility out of that is to carry some of the reporter cells that we're engineering into preclinical mouse studies, where you can implant the mice with the cells carrying these, these reporters for target engagement, and then you can identify through the mouse in vivo work whether or not there is selectivity, there is efficacy, and whether or not there is general systemic toxicity on the PLK level. So, but the process will have to include other components to really decipher the aspects that target engagement by itself is not sufficient to answer.
Makes sense.
Yep.
Yeah.
Wonderful. And we'll look forward to seeing you again, Dr. Elmar, this afternoon. And with that said, we'll be heading over to our next presentation before our lunch break by Dr. Mark.